make a couple extra explanations based on what we've been discussing. So just to reiterate, so the principal components is computed at using the gene expression matrix here, and then we're looking at the population structure of the cells in principal component space. And so the first principal component, by definition, is the linear combination of gene expression values that explains the most variation between genes. And the second component is the axis orthogonal to that explains the next most variation, and so on. So when we're looking at this plot, we're looking at cell level values. So each of the points on this plot is a cell. And so on the y-axis, we have the PC value. And on the x-axis, we have the value of, in this case, total features for that particular cell. So here, PC1 is the axis of variation that explains the most variability between cells. And that is highly correlated with total features, suggesting that total features is, as a variable, has a strong explanatory power for variation amongst cells. And so that's a bit different from this plot, which is now flipping our attention to the gene level. So if that wasn't clear when I explained it earlier, we're fitting a model to each gene with a particular explanatory variable like total features or total counts, and then we're looking at the distribution of R-squared values, so the percentage of variance explained for a particular gene by a particular variable, and then looking at that distribution. It's worth remembering when looking at density plots like these that for a density plot, it's like a probability distribution, so the area under the plot always has to be 1, and so the height of the curve is not in itself meaningful. What we're looking at here is where this distribution is situated. So is it shifted to the right towards the 100% end, or is it shifted towards the left? So if we have a very narrow distribution like the green curve for batch, then they're saying batch explains quite a similar proportion of variability across the whole range of genes. If we had a very flat curve, then that would be saying that this particular variable explains a large proportion of variability for some genes and a low proportion of variability for many other genes. And if it's shifted to the right or to the left, then that's where the interpretation in that plot lies rather than the heights of the curves themselves. Okay, are there any final questions on this part? Cool. If not, I'll hand over to Laura.
Did you go through the reads exercise? So gone through the reads, you should get plots that look like these. Again. Oh yeah, yeah. So PC1 is pretty much always correlated with total features if you haven't normalized your data at all. It's going to be correlated. Yeah, so this is what you should get for reads. Looks pretty much the same, only noisier. Yeah, you can see batch is still one of the most important ones, as well as total counts in orange. Total features a bit less important here, but yeah, total counts and batch are still important. So any questions on any of that? No. So the next section is normalizing for library size. So we're just identified important confounding factors, and one of those is always the total reads you get per cell. It's always the most important, one of the most important confounding factors. So we want to normalize out and remove that confounding factor. So there's many different ways of doing this. The reason this is a problem is because we're sequencing this on multiplexed platforms, so you get random amounts of reads being attributed to each cell. If you're just counting the number of reads per gene, you are going to have to correct for library size, but if you use, for instance, cufflinks or RSEM to get FPKMs or TPMs, they've already corrected for library size for you, so you don't need to do that. But most of the time you want to get your read counts or your unique molecular identifier counts, and then you need to normalize for library size yourself. So there's many different ways of normalizing for library size, from bulk RNA-seq, because it's a problem for bulk RNA-seq as well. The simplest is just doing counts per million. So just take the sum, so the sum across for each cell of the endogenous genes, divide by that, and then multiply by 1,000,000, which is very easy to do and very fast to calculate. But the problem with counts per million is if you have many genes that are both highly expressed and differentially expressed across your cells, then the total counts will reflect the differential expression of those genes, in which case normalizing by total counts will actually skew your normalization factors based on those genes, and will actually decrease the differential expression of the highly expressed genes and increase the appearance of differential expression for lowly expressed genes. There's many variants on CPM. So there's RPKM, FPKM, and TPM, which are all just variants on counts per million, where you also normalize for the gene length or the transcript length in different orders. Sure, I think we're going to talk about that later. So to deal with this problem of differential expressed genes, there have been many other size factor methods that have been proposed. They're all basically the same as counts per million, where you calculate some size factor for each cell and then divide the read count for that cell by that size factor. So one of the methods is called size factors from DSeq, and it's based on the geometric means. So here you're calculating the geometric means across each gene, and then finding the median median count divided by the geometric mean for each cell, and that becomes your size factor. Which you're doing here, so the median count relative to the geometric mean. The problem for this is because you're calculating geometric means, you're multiplying things together. So if you have any zeros, so if you have a 0 expression value for a gene in any of the cells, your geometric mean for that gene is going to be 0, and is thus useless for calculating a size factor, because this single cell RNA-seq, that's going to happen for most of the genes. So you're going to end up only using the top like 10 or 20% of genes to calculate your size factor, which is going to completely bias them. There's another method based on upper quartile, which is very similar. So you take the whole distribution of expression values for each cell and find the upper quartile, so the 75th percentile of that, and use that as your normalization factor. Again, we have a problem that if you have a large number of zeros in your data set, sometimes this upper quartile can be 0, and you can't divide by 0, and so you can't use this normalization for that. There are two ways sort of around this. One is to exclude all of the zeros before you calculate the upper quartile, or you can use something much higher than the upper quartile and use like the 99th percentile. But again, you're now using very small number of your genes to base your normalization factor on. And then finally, there's also TMM, which is weighted trimmed mean of M values, which basically uses one cell as a reference and then calculates the log2 fold changes between all the other cells in that one reference cell, and then trims that distribution and picks something in the middle of that as your normalization factor, to then adjust all your other cells to match your reference cell. But again, because looking at fold changes, zeros can completely screw up this method. It's supposed to be random. I think in practice, usually they just pick the first one. Yeah, it doesn't. So the final method we're going to look at is the first one that's been developed specifically for single cell data in order to deal with both the problems of counts per million, but not have all these problems with dealing with zeros that the other methods have looked at. And basically it works by first pooling cells together, so just summing up the reads from each cell, calculating the size factor for that pool, and then using linear algebra to break those pools down back into a size factor for each individual cell. So you do many, many different pools with different combinations of cells, and then break it down back into one size factor per cell, which is implemented in the SCRAN package. In order to compare these different normalizations, we're going to use PC plots, but they can get quite difficult to compare by eye, so we're also going to use calc-cell RLE, this function that I've provided, where basically you take the median across all of the cells for each gene, and then look at the expression value in this particular cell relative to the median across all of the cells for that gene, and then take the median value for, so you get one value for each gene in each cell doing that, and then calculate the median of that across all of the genes in each cell. If your cells are well normalized, that should be close to 0. If you have a cell that has higher read counts for most of the genes, you're going to end up with a positive value. If you have a cell that has lower read counts for most of the genes, you're going to get a negative value for RLE. And it's just a way of summarizing things nicely and quantitatively. Yep. You could add a pseudo count of 1 for the geometric. So the question is what to do. Can you just fix your geometric mean by adding a pseudo count of 1? In theory, you could, but that's going to completely skew because a pseudo count of 1 is different depending on the actual value. So if you have a lowly expressed gene, adding a pseudo count of 1 is going to increase that a lot, whereas for a highly expressed gene, adding a pseudo count of 1 is not going to change it very much. So it's going to end up skewing your size factors still. Yep. So again, we're going to be working with the Blishak data, and Skater helpfully has this wrapper for calc norm factors from HR, which implements most of these methods, and it also plugs into the SCRAN package as well, so we can use all of these normalizations on the Skater object from before. So if you load up the Skater object that we've been working on, and plot the PCA of the raw count, so this should be what you saw in the 2 chapters ago, just the PCA plot. And you can see here the point size is relative to the total features, and so you can see that component 1 got all the cells with large number of features over here and small number of features over there. And if we calculate the cell RLE, we can see a very broad distribution, indicating poorly normalized data. If we calculate counts per million using this command, this is automatically calculated in Skater for us in the express variable. That'll give you the log 2, I believe, counts per million. and then plot the PCA plot, and we see quite nice different groups appearing for each of our different individuals. So this is all one individual. These are batches for a different individual, and that's a third batch for an individual. And we no longer have a huge bias based on the total features. And if we calculate the cell RLE, it's now almost all completely close to 0. And we can do the same thing for TMM. Here we calculate the normalization factors, and then we plot the data using scatter. And it's all nicely inbuilt. And this is not as nice as the counts per million. You can see it's all kind of messy. We have gotten rid of most of the relationship between total features and component 1. However, it's not too obvious anymore, but it's all kind of messy going on. The different individuals are not too distinct. And calculate cell RLE, and it's shrunk a lot, but there's still some there. And then we can use scran. So scran is a bit more complicated. So this first command will cluster the cells so that we have cells with similar read depths in each group. And then for each of those groups, it calculates different pools and calculates the size factors after pooling each of those little groups. It just makes the method faster and more accurate. So we first cluster them based on the read depth, and then compute the size factors, and then do the normalization. And then plot it. And you see we get something close to counts per million. Again, looks pretty good. Each individual is very nice and distinct, although particularly for this individual, you can see batch effects where each different batch is quite separate. And then if you look at the cell-wise RLE, it's very flat again. Yep. Yeah, so okay, so the question is how much, so for questions that for screen we can see that we've got this window size parameter, and how much does the method depend on that? In our experience, it doesn't matter that much as long as it's more than like 10 or so. It doesn't make very much difference. I think it mostly affects sort of the runtime rather than the actual values you get at the end. Right, and then we did size factors from DSeq. And again, see this is not as good as the counts per million methods. And do the cell RLE. And see we've got quite a few outliers up there, but otherwise that looks reasonably good. And there's upper quartile, which looks very, very similar to the size factors method, although it has a bit higher RLE remaining at the end. So up until here, I've just been looking at size factor-based methods. There's one other method to correct for library size, and that's just a downsample. So you have a you have your read counts for each cell, so you can just downsample those read counts until they're all the same total at the end. So the benefits of this is that, so using the other methods, you're just dividing by a value. So obviously all your 0 values are going to be completely unaffected. Anything that was a 0 is going to still be a 0, and anything that wasn't a 0 is still not going to be a 0 at the end. So the number of zeros has not changed at all using the other methods, whereas downsampling will change the number of zeros. Because downsampling, once you get below 1 read, you're now going to have a 0, new zeros appearing where there weren't before. However, the problem with downsampling is that because it's a stochastic process, if you downsample once, multiple times, you'll get slightly different expression matrices at the end. So then you can very quickly end up having to do all your analyses multiple times for multiple different versions of downsampling. And just sort of explosion in the number of analyses you have to do. So here's a function for downsampling. So here we input our expression matrix. First we have to calculate which cell has the fewest number of reads or UMIs. We do there. And then for each cell, we take the ratio of the observed, the minimum library size versus the library size for this cell, and then just do a random binomial drawing using that probability. And that will renormalize all your cells to have approximately the same total read counts at the end. So this is how we can apply it using Skater. And just do that function call and then plot that. And it actually doesn't look as good as the SCRAN and CPM methods. It looks closer to the UQ upper quartile and size factor methods. However, when you look at the RLE, it's completely gotten rid of all biases in the RLE. But it leaves a lot of noise in the data because it's a random sampling. So we're going to come back to normalizing for gene and transcript length, which are different variants on counts per million. So first we have reads per kilobase per million and FPKM's fragments per kilobase per million. It's just single end versus paired end sequencing, where first you basically do counts per million on your data, and then after you've done counts per million, you then normalize for the gene length. Whereas for transcripts per million, you first normalize for the gene length, and then you do counts per million. So these are interchangeable. If you just take FPKMs and reapply counts per million on top of it, you'll get transcripts per million. However, we're going to try applying this to our data set, but it makes absolutely no sense to do that because we have a UMI-based data set, and the UMI is only located at one end of the transcript. So the sequencing that has been done for this data only sequences that end of the transcript. So if we go down here, I have a nice plot. You can see that there's no relationship between the length of the genes and the total counts or the average expression level, where we see for each gene. That's because we're only sequencing one end of it, so there's no relationship there. If you were sequencing the entire length of the transcript, you would see a positive relationship here, which would indicate you should normalize for gene length. However, I'm going to put a lot of stars at the end of this because even for whole transcript sequencing, we saw earlier that you get a very strong bias towards the 3' end. So the actual length that you're sequencing is not the same as the read length that the transcript length you'll find in the database. So it gets very complicated to correctly normalize for gene length in this data set, in these data sets. So we sort of recommend you use cufflinks or RSEM or Callisto or some mapping algorithm or quantification algorithm that will actually look at where your reads are mapping when it's calculating the transcripts per million, as opposed to trying to just, after the fact, normalize for it. But if you want to try doing it after the fact, these are the commands to do it. So first we have to go get the gene length for each gene using this command to access Ensemble Biomart. And then we have to make sure we're only considering those genes where we have annotations in Ensemble, which we're doing here. Then we calculate the gene length according to Ensemble, and then we plot the two to see that there's no relationship. And then we can use that effective length to do transcripts per million and calculate FPKMs, again, using Skater that are already implemented for us. But you can see that you won't realize that it's completely inappropriate to do this using PCA. This is because PCA is only looking at differences between cells, and because you normalize for the gene length across all of the cells in the exact same way, it doesn't actually affect the differences between the cells. So the PCA plot will look exactly the same, or mostly the same, and you won't be able to tell that you've done something silly. Whereas TPM, because you're doing the normalization for the gene length first and then doing the counts per million, it will have differences because you're essentially changing the weighting of each gene based on the gene length. So then we have an exercise for you to do the same analysis with the read data from this data set. So I'll give you 10 minutes. Oh, okay, so we're going to have a coffee break as well. So you'll have 15 or so minutes to work on this exercise and an extra 5-10 minutes for a coffee break as well. So work on the exercise, have coffee. Yeah, and we'll continue again in a little bit.